welcome to today's show. The American Society for Gene and Cell Therapy's annual meeting is only a few weeks away. The abstracts were just released, and in today's show, we're going to dive into what are some of the top abstracts at this year's annual meeting. It's really impressive when you look at the volume of abstracts that were submitted. This was the largest ever number of submissions to the meeting. There was a 13% increase in abstract submissions in total, which really, I think, illustrates the scope, enthusiasm, and maturing of the field. And I'm joined today by two special guests, experts in the field of cell and gene therapy. Dr. Walsh Martin has spent over 2 decades in biotech, where she has held program leadership and management roles across a range of disease areas and has worked directly with numerous teams from candidate nomination to the clinic in a range of therapeutic modalities, including gene therapies, cell therapies, biologics, RNA, and nanoparticle delivered nucleic acids. And joining Dr. Walsh Martin is Lynnea Olivarez. Lynnea is a life science communications practitioner and incoming ASGCT Communications Committee chair. Lynnea specializes in integrated PR, digital and social media, and employee engagement initiatives. She's also recently launched the Ticket to Biotech, a global networking and professional development community specifically for biopharma communicators to address the challenges in this highly specialized profession.
Let's maybe jump in and start with the Presidential Symposium. I'd love to get your thoughts on the top abstracts there. So Emily, do you want to get us started with the first abstract?
Sure. So, I mean, this one really I'm very excited about because I believe I haven't fully parsed everything, but I believe this may be the longest duration of follow-up in our abstract book this year, which is absolutely amazing. So they have a single patient that was treated with HST gene therapy for metachromatic leukodystrophy that's been followed up for 12 years at this point. And on average, I think the follow-up was around 6.5, almost 7 years, which is pretty amazing data. This data is super important to us as a field. Long-term follow-up really gives us a better sense of how well is our design, because with most of the design characteristics that we imbue these cell and gene therapies with, when we were in discovery, it's really the durability question that we're all sort of waiting to understand the outcome for. And so I'm very excited for this abstract in particular because we're starting to see that long-term data roll in.
So Emily, for our listeners out there, this is the Orchard Therapeutics drug, Lynnea, and which had really impressive overall survival effect and preservation of motor function and cognitive skills in most MLD patients. Lynnea, what were your observations on this abstract or your thoughts?
Sure. So before I dive in, I actually wanted to take a step back and make a comment on the overall Presidential Symposium suite of abstracts this year. And it really struck me as a microcosm of all the hot topics happening in CGT right now. And I'll come back to this in a second with my additional thoughts. But first, as Emily was saying, you've got more data to support this recently approved gene therapy that, by the way, broke the record for the most expensive drug in the world. You have in vivo cell therapy, and dare I say allogeneic, 1-year NHP data, and you have early proof of concept for a new delivery modality, like all super exciting. And I don't know that I could have predicted this even a few years ago at coming to an ASGCT meeting. And so I think we have a really exciting slate coming up. But to go back into this first abstract, To give a little bit of background on the way I was looking at this. So for those who may recall, Orchard made waves with this $4.25 million price tag, but they actually said in their pricing announcement that Lenmeldy is launching with the longest duration of follow-up available at the time of approval for gene therapy in the US to date. And this presentation at ASGCT is actually their first scientific presentation post-approval. So I'm personally really curious to see how they use this stage and use this opportunity to support that case and further their narrative.
Yeah, because I think the real risk with, our field in general, right, is that we, especially with diseases that are devastating, like MLD, where, the fatality rate and just the rate of decline is just devastating. The risk is that the mild benefit, perhaps, of a gene therapy doesn't sort of get the patients over the hump, or gets them to a new hump that basically doesn't really address not just a lack of fatality, but also just a quality of life. And so that's what I'm really excited about in this abstract, is seeing that long-term data, seeing how these the patients are doing today. They comment in the abstract about a lot of the endpoints that they were able to reach with pretty, shocking p-values. But I think it'll be really good to have them walk through the qualitative improvements that these patients are seeing, so we really understand. Are there additional areas where we could continue to improve these patients' lives, and how might we do that with some of the new tools that are coming out? But importantly, long-term data is gold in this business, and I'm really, really excited to see it being highlighted as the number one presidential symposium, at least in order in the abstract book.
Yeah, it's really, really interesting looking at the most common adverse reactions, mainly hematologic, some stomatitis, rash, some viral infections, but really very well tolerated. So yeah, agree with you, Emily. We'll be paying close attention and looking forward to the presentation and looking how these patients are doing. Of course, when you're talking about a young patient population in pediatrics, we want to make sure that that the long-term safety is solid. So let's, if we can, let's move on to the next abstract, where this is actually a preclinical abstract, but it's in vivo generation of both CAR T cells and CAR NK cells using a CD7 targeted lentiviral vector. Emily, what are your thoughts on this abstract?
Yeah, so this one I'm also super excited about because it solves one of the key problems, right, that the field faces today, which is oftentimes in CAR T oncology studies, the challenge of apheresis, lymphodepletion, manufacturing failures, et cetera, everything that's between a patient going on to a study and then eventually receiving the therapy. It's a long process. It's an arduous process, and it's a process that can have failures at any point in time. So finding paths to remove some of those obstacles to benefit is super important for the field. And this is exciting data, again, preclinical data, so to be determined how well this translates into the clinic. But they're using a novel lentiviral vector that's targeting T cells as well as NK cells, and it's administered intravenously. So there's no apheresis, there's no ex vivo manufacturing process, there's no lymphodepletion and then administration. You're basically jumping straight to the dosing. It was really interesting to me their comment that they're not seeing hepatocyte transduction. So that'll be really cool data to see on the big screen. But again, if this continues to show the same promise in the clinic, this would be really, really interesting for the field.
Yeah, I echo that, Emily. Granted, I think this study was in a small number of animals, but just to quote for a moment, they said that the depletion of peripheral blood B cells in vector-treated NHPs was evident as early as day 4 post-administration. And that systemic B cell depletion was confirmed at necropsy 1 year post-treatment. And so even for animal data to see that they have that a year out, and on top of that they generated the NK cells all in vivo, super impressive. I also am really excited about this no off-target transduction seen in the hepatocytes. And I saw they noted in their abstract that they have their planned FIH study later this year. So this is certainly an exciting precursor to that. And then just another note I wanted to mention. So this is coming from the company Interius, and notice that they're based in Philly. So Philly is continuing to shine as a rising CGT leader. So really cool to see some more data coming out of the Philly corridor.
Well, those great points. I think that for me, this in vivo generation of CAR T cells, endogenous CAR T, is fascinating. And obviously, you've already mentioned the manufacturing benefits and taking that step out. I know the abstract mentions that they're looking at this across a broad range of hematologic malignancies, and it shows promising results in B-cell lymphoma. But I think for me, I'm really going to be looking at side effect profile, because I think the whole promise of in vivo generation of CAR T is lower rates of ICONS. That's the immune effector cell-associated neurotoxicity syndrome, cytokine release syndrome, and GVHD. So I think we're really looking to see what that looks like. And I think for me, this is sort of I still need to see the data on this one. So
yeah, well, and I totally agree with you. I think the big unknown here is how well can you titrate in the clinic, so to speak? How much variation are you going to see patient to patient for a given dose level? And there the devil's going to be really in the details. And I don't know that you could even hope to model that preclinically. So we're just going to have to wait.
Great point. Let's move on to the third and final abstract in the Presidential Symposium, which is a novel vector being used to target the epithelial cells in the lung for cystic fibrosis. Emily, please do tell us about this one. This is fascinating.
Yeah, so Boca virus for the win, right? So this is a new vector. It's got a much bigger payload capability than AAV, so that's a plus. And this is coming out of Carbon Biosciences, and their first, foray. They're loading it up with the CFTR, cystic fibrosis transmembrane receptor, to try and treat cystic fibrosis. And for those who aren't super close to CF therapies, right? There's been amazing progress for in small molecules for patients who have a CFTR that's not completely deleted, right? Where there's a big unmet need is for patients who basically have no functional copy, no expressed copy of the CFTR that could be modulated by some of the small molecules that are available. Now, the other part of this, though, is that the bar is you want to see efficacy that matches those small molecules, because what I think has been amazing from the small molecules that are out there is the level of benefit that people are seeing in CFTR patients, or cystic fibrosis patients rather, is pretty amazing, given how challenging this disease is. So this group is presenting the gold standard, the CF ferret model, which is great to see. What they're showing is that the Boca virus can transit the mucus layer in those CF ferrets, which is great to see as well, and transduce lungs. What It's unclear if they're going to share is any durability data, because that's, the devil's in the details on that. We know that lung cells actually turn over pretty quickly. And so you have to be able to get to some of the longer lived lung cells, or you have to be able to redose. So those are the big questions for, a viral vector approach like this in CF. But I'm excited to see the progress that they've made, and especially excited since it's some great ferret data.
Yeah. And from a corporate perspective, so I was looking back at their history, and they launched almost 2 years ago coming out of stealth. And so for me, this is kind of a mic drop moment. Like they've been kind of quiet the past couple of years since they announced their initial funding. Carbon has, and now they're coming out with an abstract in the top 3 in the Presidential Symposium at one of the premier conferences in our sector. And so as a company, I think that they must be super jazzed about that. And it's great validation for their initial value proposition, right? They're talking about developing this delivery platform leveraging their novel parvovirus vector library, which, as Emily said so eloquently, is a new vector modality, and in theory can solve a lot of these common challenges that we know, unfortunately, all too well, including payload size, delivery, so forth, and to see some such initially promising data in this gold standard model, as Emily says, is certainly very, very encouraging. I'm also really curious to see how they, as a company, talk about this, given all of the additional, frankly, really cool work happening in the CF field right now. There's a lot of competition out there to get that genetic medicine for CF patients, which is awesome for patients. But it's pretty cool to see this new modality making waves at this conference.
Yeah, me too. I think I'll just close on this section by saying that, I agree, as Emily mentioned, this gene delivery mechanism with the Boca virus addresses the limitations of cassette size for traditional viral vectors. To me, still the immunogenicity is a key factor that we don't know the answer to, but we'll see from the presentation what they share there. Of course, it's preclinical, and we may not know until the first in human studies would start, but nonetheless, very promising development here. I'm looking forward to seeing the data.
Yeah. And I mean, obviously, I think that, again, the devil's in the details, as you mentioned, Chris and Lynnea, but, so this data is intratracheal administration as opposed to a nebulized administration, which is totally understandable for where they are, in their path of discovery. But there's a lot more steps. There's, making sure it can be nebulized. There's a question of repeat dose, repeatability of dosing, or durability of dosing. What are they seeing? And so very exciting initial data, excited to see what happens at future ASGCTs for this project.
Well, good. That was a good discussion, good overview of the Presidential Symposium abstracts. I'd like to go a little deeper into the abstract book and look at the clinical trial spotlight, go a little deeper into base editing, prime editing data, and also deeper into some of the other therapy areas that folks attending ASGCT could expect. So I'd like to kick things off with another area that's white hot right now, which is the personalized DNA vaccines. So we know that there's a new generation of cancer vaccines that was center stage at AACR just a few weeks ago, and where Jynneos Therapeutics reported data in advanced liver cancer with really good efficacy with its personalized neoantigen DNA vaccine, Genos PVO2. Certainly their data compared very well compared to just pembrolizumab alone. The cancer vaccine is tailored against neoantigens identified in a patient's tumor that may enhance responses to PD-1 inhibitors through induction of tumor-specific immunity. So at the annual meeting this year, we'll see that the company will report results from a phase 1/2A trial evaluating Genos PBO2 encoding up to 40 neoantigens. It's co-administered with a plasmid-encoded IL-2 in combination with Pembro in patients with advanced HCC. Emily, any thoughts on this one? I mean, this is a hot space with Moderna. I'm going to come back to that in just a moment, but your thoughts?
Yeah, I haven't been following this one as closely as maybe I should, and it'll be exciting to hear them speak about it, at the meeting. I think the challenges really are with complicated therapies like this is basically, are you going too deep? Are you going too broad with the neoantigens that you've selected? And how does that sort of pan out when you go into increasingly bigger patient populations? That said, it'll be really interesting to hear them talk through the data that they have right now, including it looks like 3 radiographic complete responses. So definitely devil's in the details, but excited to see some progress being made.
Yeah, and I'll just mention, because there was a lot of debate last year at last year's ASCO when Moderna presented the phase 2B Keynote 942 trial with their product mRNA 4157 with Pembro in melanoma, in advanced melanoma. And their synthetic mRNA codes for up to 34 neoantigens. So Genos PBO2 from Jynneos Therapeutics is sort of in that same, 30 to 40 sort of neoantigens. We don't know yet as a field how many neoantigen targets do you need to target, right? And so that to me is the interesting question.
Yeah.
So I'm looking forward to seeing that T cell response data for Genos PBO2 plus Pembro combination at the meeting. I'll just move on to abstract 9, which talks about the safety and efficacy of AB1002 gene therapy in patients with advanced heart failure. The sponsor here is Asclepios Bio. And here we know that patients with heart failure often have abnormal intracellular or membrane calcium signaling with increased levels of protein phosphatase. So AB1002 is a novel cardiotropic liver detargeted capsid, AAV228, designed to overcome this poor membrane transduction of calcium. And this abstract actually is interesting. It reports that AB1002 is well tolerated and has positive efficacy outcomes in patients with non-ischemic heart failure. So I think it's fascinating as we're starting to see gene therapy trials go outside of the traditional sort of, monogenic diseases, the rare diseases, oncology and hematology indications. Now we're going into these big indications, more prevalent indications like heart failure and cardiovascular disease.
Yeah, no, that's one thing. I'm glad you brought that up, because this abstract book is sort of filled with sort of new directions. I saw a number of osteoarthritis, for instance, programs, including actually one that I'm helping to present on Saturday. But there's a lot going on this year with these more common diseases, and also diseases where you're not doing a gene replacement, but you're nudging things, if you will, biologically. So I think the key thing here is, these are studies where numbers matter, right? Because these are generally larger patient populations, and so really understanding patient experience across, instead of tens of patients, more like hundreds of patients, becomes when you really start to understand if you've got a therapy that's going to have the right clinical equipoise, if you will, for treating a common disorder in this way. So more to see, more to discover, but I would definitely encourage folks to attend at least one of these more common disease presentations, especially if they're coming from rare disease, because I think the more we think through as a field how those two things differ and what sort of expectations we have for each, we're going to be getting ahead of some of the challenges that are probably yet to come.
And Emily, just sticking with the theme for just a moment here on cardiovascular diseases and dyslipidemias and so forth, there's a study that popped to the top of my list in this year's abstract book, Abstract 381, which is for Verve 101. Now, this study popped to the top of my list given Verve's recent press release on April 2nd that announced the company's actually discontinuing development in Verve 101 because of they identified in a patient elevation in liver enzymes. And just to remind our audience, Verve 101 is an in vivo base editor composed of an mRNA encoding an adenine-based editor and guide RNA that's packaged in a lipid nanoparticle. So it's designed to permanently inactivate the PCSK9 gene in the liver with a single base pair change and lead to durable reductions in LDL. So they previously reported positive efficacy data from Heart-1, their clinical study, their phase 1b clinical study. But Emily, you're well aware of this data. What's your read on this?
Yeah. So, it's, again, this is, what they're trying to do is knock out PCSK9. There are a lot of therapies that do this in different ways that are already marketed. There are antibodies for PCSK9. There's inclisiran from Alnylam that is a siRNA targeting PCSK9. I think the rationale in this approach is it's going to be important to overcome some of the challenges around patient compliance, et cetera, to really have a durable, sustained benefit for patients who suffer from dyslipidemia. I think that said, there's a strong sort of risk benefit that you have to keep in mind with an indication like this. And so I think right now what Verve's going to do is they're going to pivot to a new LNP, it sounds like, and pursue that in the clinic. It'll be this new LNP, I think, is expected to do two things. One is it may reduce the risk of LFTs, according to their, I think, preclinical data that they've mentioned publicly. And then secondly, it has a targeting moiety on it, which could allow it to transduce potentially more efficiently in these patients through two different receptors, basically the usual receptor and then a GalNAc receptor. And so, I think this is, an unfortunate setback, but it'll be important to watch and see how this turns out. There, in theory, could be other reasons why they saw these LFT elevations, but this is an easy one for them to sort of assess clinically and figure out. So we'll have to just watch and wait and see how that turns out. Yeah,
I agree, and I think that I'll be looking at the data because I think it does provide proof of concept for in vivo based editing of this PCSK9 gene. And I'll be looking at that sort of across patient population or across ancestries to see what takeaways we can take from this study as the company shifts to their new LNP Verve 102.
Yeah. And there's also a case to be made, right? Like, so, when you look at the inclisiran data, right, they're able with this siRNA to really knock down pretty strongly the PCSK9 gene. And yet for some patients who have rare genetic forms of hyperlipidemia, it's still not getting them into that normal zone. So I think for me, I'm also interested to see where Verve goes next in terms of, additional targets beyond PCSK9, because this may be a case where it's not going to be a single gene that's going to give the full benefit to some of the rarer dyslipidemias out there.
So let's move from prime and base editing, and I'd like to just look at a couple of abstracts from the targeted gene and cell therapy track. For many attendees, many of us in this space who have spent many years working in oncology, hematology, the holy grail in this field, right, is how do we take the potential of what we've seen with CAR T and hematologic malignancies and move it to solid tumors? So for me, abstract 415 is really interesting and should get a lot of attention because that abstract's about KRAS mutant gene editing that has shown to abolish tumor growth in vivo and overcomes the acquired resistance to KRAS G12C and KRAS G12D small molecule inhibitors. So we know there was this race by two other companies racing to develop the KRAS targeted therapies, and they're now marketed. And when I look through this abstract, this is potentially a new strategy combining in vivo gene editing with tumor selective nanoparticles again to inhibit tumor progression by directly targeting mutations in codon 12 of the KRAS oncogene.
So I think what's interesting to me is that they mention that they do 2 administrations of the therapy to abolish the PANC1 tumor growth in the mouse model that they are exploring. And so it'll be interesting to see if they present like single dose data versus 2-dose data. I think, sometimes when you're at this early stage of development, understanding sort of the interventional strategy becomes the key part to unlocking, your path into the clinic. So getting a look at that data, seeing what, the 2 administrations does, maybe hopefully they're showing versus a single administration, will be, I think, really important to see how effective this could be and what path could be sort of pursued.
Agreed. And I'm looking forward to seeing this proof of concept data and how ADGN complexes can be applied to driver mutations in solid tumors, because this could apply to a number of GI cancers, lung cancers, pancreas cancers, and so forth. And while we're on the topic of GI cancers, the final abstract for me in this group is abstract 141, which is a novel CAR T cell therapy. This is a clinical stage phase 1/2 dose escalation study for GCC19 CAR T for patients with metastatic colorectal cancer. So here's another solid tumor. Metastatic colorectal cancer has not been considered an immunologically sort of hot tumor. So I'm really excited to see this one. Again, Emily, I know this one. What are your thoughts on this?
Again, we're starting to see a lot of things coming into the solid tumor space, and I know that there are a lot of different approaches being pursued, and definitely we need to find a path, right? So it'll be very interesting to see the side effect profile for this one, especially given that you're trying to target the colon and whether or not that changes basically the side effect profile, because you'll have more activated CAR T cells in the GI tract. But it'll be very interesting to see the progress that they're making, especially, I think they have some stable diseases and some partial responses. So important data to review.
Yeah, and I'm looking forward to, because the abstract mentions that the authors intend to present updated safety and efficacy data from the clinical study, from their dose escalation study, during the meeting. So let's I'll be on the lookout for that. And then I would just say, as we conclude this abstract preview, the final 2 abstracts that I propose to mention really fall in the bucket of sort of upcoming regulatory milestones, because Rocket Pharmaceuticals has a PDUFA date in June for its leukocyte adhesion deficiency 1 gene therapy, lentiviral gene therapy, and they've got planned U.S. and European regulatory filings in Fanconi's anemia later this year. So if we start with abstract 245, the company is presenting data for its lentiviral mediated gene therapy RPL102 for Fanconi anemia, and that they've actually showed that it's not associated with insertional mutagenesis. So Emily, very well, when you're talking about lentiviral therapies, gene therapies, kind of all the issues and challenges there with insertional mutagenesis. So I'm looking forward to this data, because they're going to be presenting information on that. So what is your read on this?
Yeah. So again, this ranks among the longest term data in the abstract book this year. It was when I went through and did my clever search for years on study. This one definitely popped up. That said, I think that things like insertional mutagenesis and clonal predominance and that sort of thing that happen, they do happen quite a distance from dosing. And so right now they're reporting data where the mean follow-up was 3.5-ish years, and the range was, up to 7.5 years. But, others have experienced those kinds of findings right around 5 years. So it may be still early days for their data, but will be important to sort of watch and see what happens.
Yeah. And then the final one is abstract 246. And this is the autologous ex vivo lentiviral gene therapy for pediatric patients with severe leukocyte adhesion deficiency 1. And we know that LAD, or leukocyte adhesion deficiencies, is a disorder caused by mutations in a gene that codes for a beta-2 integrin, CD18. And this is an essential membrane-bound protein that enables neutrophil binding, extravasation, and migration to inflamed tissues. So we know that these young patients with severe LAD1 have frequent bacterial and fungal infections and high rates of pediatric mortality. So this is interesting. As I mentioned, this one is one that's has a PDUFA date of June, no AdCom. So what do you think we're going to see here, Emily? This one will also be interesting to watch. I am less familiar with this indication, and so, but this was definitely on my list to attend and view, because again, we've got some patients with reasonable amount of follow-up, so greater than 2 years of follow-up. Again, may not be long enough to see, insertional issues should they arise, but certainly should be long enough to know the level of benefit that these patients are getting from the therapy. Well, great. One final question for you both before we conclude. And I know that you're all veterans at attending the ASGCT annual meeting, and I thought maybe you could share your tips and tricks. How can attendees maximize their experience in the annual meeting that we'll all be at in a few weeks? Actually, I'm going to make Lynnea go first. Okay. Lynnea, I have to say, what I love about Lynnea is she gets the big picture and the zoom in. So I want to take notes while she speaks. Good.
Well, I this might go without saying, but I really do think it's worth spending some time, whether it's on the airplane or even before you jump into the convention center on that first day. It's really worthwhile spending some time before you go to identify the topics and sessions that are priority for you, because unfortunately, as much as you will try, it's really impossible to see it all. Unfortunately, it just is. And so I think the sooner you accept that, the more you're actually going to get out of the conference. And Emily mentioned a couple of fun ways to search through the abstract book or the great program site online, whether that's through disease area, technology, but you can also look by people too, right? If you have a hero or heroine, someone who you're just dying to hear an update from, you can search by their name as well and make sure you go to catch them. Another tip I have is to follow the meeting hashtag on LinkedIn. Or also on X, but I think LinkedIn's becoming more and more popular every year. And so I think that's hashtag ASGCT 24. I'm also going to say hashtag ASGCT 2024 in case I have that wrong, Alex. But I think it's definitely worthwhile looking at that. I can tell you from personal experience, I followed that and have seen some things pop up that maybe I had missed in my pre-planning. And it's don't miss the cool swag too. That it may not be the most scientific advice out there, but last year I think there was like a macaron painter or something. Like there are some really cool things, and ASGCT, the conference as well, has some neat swag too at their booth. So I think a prime opportunity to take a look at that is at the welcome reception on Monday. But I know that there are a lot of different times throughout to go check that out and snag your swag. And then I think my last big tip is to make sure you get to the big talks early to snag your seat. It's I mean, in our field, these are rock stars, right? There's a lot of people vying for these seats. So it's definitely worth it to go and to hear the whole thing. And take the time to network, to say hi to people maybe you haven't seen in years. Those lunchtime dates are just as important as maybe seeing that poster or that talk.
Yeah, so I'm just going to definitely echo that. So I was going to start with comfortable shoes, actually. So because there is a lot of walking around as well. You know, if you can do a buddy system, if you have a friend who's going and you guys can divide and conquer for some of the concurrent sessions and agree to take good notes for each other, I think that's also a great way to sort of extend your reach, if you will, and see a bit more. My other patented trick is to have the concurrent session I'm most interested in up on the app. So I know when to bolt from one room to the other. And I usually sort of check out to see how far the rooms are from each other. And again, the comfortable shoes come into play for making the quick, hasty getaway between meeting rooms. The other thing is, it's going to be too much and it's going to be overwhelming and you're not going to see everything you want to. And so I would say if you have your checklist of your must-haves and your nice-to-haves, try to network. And if you see someone from the institution of one of your nice-to-haves and you're pretty sure you're not going to be able to sprint to that talk, connect with them and say, hey, I'm going to miss the talk, but do you mind if we follow up by email afterwards? This is a meeting where you absolutely can walk up to anyone and say, hey, my name is Emily, and I'm really super psyched about what you do. I'd like to hear more. Can we connect? So take advantage of that. This just in, it is ASGCT 2024 is the hashtag. That said, search both because invariably no one does it all on the same hashtag. So that's another tip. And I think the last thing I would do is also plan to attend not just this annual meeting, but also my favorite meeting, which is the Policy Summit in September. Because I think when you think about all the steps from discovery to actually delivering a commercial product to a patient, you need the full arc of that. And that's something you can get educated about at the Policy Summit. So save some energy for September as well.
Well, great. Thank you both. This was a great roundtable discussion. I'm looking forward to seeing you in Baltimore in a couple of weeks. And thanks for all the great insights.